and share my screen. And let me go to Canvas. So if you click on Home or Modules, this will come up. The recordings are right there, Panopto recordings. And they became available sometime last night, meaning they're not available right after you, right after the class. It takes them a while. And I don't know if somebody is physically making them available or not, but hopefully they will be available sometime after the meeting. And then I didn't know this. I need to go in. I just learned this, I don't know, several hours ago, that I need to go in and edit them and make the captions available. Most of you aren't using captions, but I do have one student who is using captions, and so I need to do that for each video. And I don't know if I have to do that before it's published or after, but it doesn't matter. I'll figure it out. So to make them available, there is a little bit of work, but this is where they will be. They are also on my YouTube website, and I will do that at least for the time being, so that you'll have access to them in two places. And you click on the videos of class, and it tells you where they are, and click on this link and you can go to the YouTube site. Actually, I want to do that. I wanted to see if the captions on YouTube are their own captions, or if they are Panopto captions from Yavapai.
Can I ask a question? Is there like somewhere where you have notes where we can follow along with what you're teaching every day?
Yes.
Because I tried to... Okay, where do we find those to print them out?
Recording. I'm going to share the screen. I warned you before, I'm not sharing my screen. I see somebody's done a chat. I don't see... Oh, this is the lab, messages. Yeah. So if you... Oh, that's fine. You're just asking other students to study together. I think this is Yahoo's captions. Anyways, I wanted to look at that. So I've got the videos in two places, and I'll continue doing that at least for the time being. Why I want to put it at YouTube is I'm not sure how long I can keep them on Panopto. So I'll want to put a copy, my own copy, on YouTube. And the way it's set up now, I don't have a copy for myself, but that's okay as long as I have a copy on YouTube. All right. Somebody else was asking a question. What was that question?
I wanted to like have notes of what we're learning every day, and I cannot find where you do that. Print those out.
Go to the modules or the home. I think that's the same link, actually. Yeah, it is. And scroll down to the lesson. So I've got the overview and the objectives that just disappeared. And then the study guide will be the notes in a text format. So this will be the same material, but in a different context. Okay? And so you can add notes to these if you like. After the lesson is done, I will make the slides available to you, but I want you to write your own notes, so my slides will not be available to you until after we've covered the chapter. And the reason why I'm doing that is it's well known from studies that students, if they just print out the notes and then study from them, they won't learn the material as well if they take their own notes and study from their own notes. There's something about your own notes that help you remember the material. And I don't know exactly what that is, but it's because it's your notes, you will learn the material better from your own notes. So I want you to take your own notes, but I have notes available to you. And the text format of the notes are available now. Another thing I need to talk about is the link to the lockdown browser is now working. That was actually quite a chore. It took me an hour to figure it out and what to do and how to replace it. But this link now works for Yavapai. All it is a different ID. And I didn't know this lockdown browser has a different lockdown browser, or a link at least, for each college. And anyways, I made a mistake and I copied from my Clark College website and put the same link there, and that's why it wasn't working. I've also looked into, I guess I got to show you that. I looked into the length of the class, and on Tuesday and Thursday we have a really long session, and this is correct, that at Yavapai on Monday and Wednesday we have a short lecture from 1 to 2:15, and then the lab starting at 2:30. I will probably break around 2 o'clock and then come back and continue today at 2:30. So you will have a fairly long break where you could go and eat or do something. We can talk about what you want to do about that in the future. We could also continue going on and then end, I don't know, like a half an hour early. But on Monday and Wednesday, no, sorry, Tuesday and Thursday, we have a really long session from 1 to 3:45. Okay? Any question about any of that? All right. If not, let's scroll down to the bottom. Take a look at what we're doing today. We are continuing Chapter 1, and we have such a long session, I have a feeling we'll be close to finishing, maybe not quite finishing, but I have 3 days for Chapter 1, and we have such a long day today, I'll check and see if we'll end Chapter 1 early. There is no lab today, it's only lecture. All right, I'm going to close this down and then move on to where we were. We were talking about microorganisms last time, and they come in various shapes and sizes. They are all small. All microorganisms are small, but they are different shapes. Microorganisms play many beneficial roles on the planet and for us. We've discussed a few of them. Microorganisms decompose organic waste. And then I mentioned this is more than just what we flush down our toilets. It is also when a tree dies on the planet Earth, it will be decomposed largely by microorganisms. It is true that mushrooms can also decompose trees, but most of the decomposition is done by microorganisms. Microorganisms are also producers in the ecosystem. Producers are organisms that make food for the rest of us. And even if you eat cow, meaning beef, the cow eats grass, and grass is a producer. So the cow got its food from the producer, and then you got your food eventually you got your food from the producer. So producers are extremely important in the ecosystem, and producers all engage in either photosynthesis, which is most producers, and a few producers engage in chemosynthesis. Green plants engage in photosynthesis, and they are the producers you're familiar with. But there are microorganisms which are producers, which do engage in photosynthesis, but there are some that engage in chemosynthesis. And chemosynthesis is they get their energy not from light, but from a chemical. And we'll talk a little bit about chemosynthesis later in this lab. Microorganisms are also involved in the nitrogen cycle. We talked about that. And they are involved in other cycles. We'll talk about that in a little bit. So they are important for recycling nutrients on the planet Earth, like recycling nitrogen, moving it around from one place on the planet to another place. Microorganisms also produce a large number of, chemicals or materials for us. So for industrial chemicals, we use microorganisms to make ethanol and acetone. If you don't know, the ethanol that is in your car is made by microorganisms. And usually, let me see, is it the winter or the summer? They put ethanol in the gasoline in the United States. In one of the times of the year, and I don't remember if it is in the summer or if it's in the winter, but they do one of the times of the year. We use microorganisms to make insecticides, and this is an alternative to man-made chemical insecticides. We use microorganisms to ferment many of our foods, the fermented foods. Like we use microorganisms to make vinegar, to make all forms of real cheese. What I mean is not a processed cheese, a real cheese like cheddar cheese or Swiss cheese or some other cheeses, whatever the other cheese is. We use microorganisms to make bread, as long as it's leavened bread. And then we use microorganisms to make beer. Microorganisms also make other products for us, like products used in manufacturing. An example is the enzyme cellulase is made by microorganisms. We use microorganisms to make antibiotics for us. Penicillin G is an antibiotic that is made by a microorganism. And we'll talk a little bit about that. Many other antibiotics are also made by microorganisms, like, oh gosh, I forgot the name of it now. Ampicillin is made by a microorganism, and then humans chemically modify it. But it is first made by a microorganism. We have microorganisms which make other products for us, such as products used in medicine. For example, all human insulin that diabetics take is made by microorganisms now. Initially, insulin was made by humans, and I don't know how they derived that, but they isolated it for humans and then purified it and gave it to diabetics. Well, that was a very inefficient way of getting it. And now we grow the insulin in a microorganism. We put the gene in the microorganism, and then the microorganism makes it for us. Any question about any of this? All right. Modern biotechnology and genetic engineering also use microorganisms. Now, biotechnology itself mostly does not use microbes, but it does use microbes and other organisms to produce foods and products for human use. And the use of biotechnology is not something new. It is centuries old, like the fermentation of cabbage into sauerkraut. That is biotechnology, which uses microorganisms, but it is centuries old. And the making of other products, like we developed corn, it's centuries old, biotechnology. Genetic engineering is a new technique of biotechnology, such as adding genes to the organism so that the organism will produce something that we want. Genetic engineering is new, and it is a branch of biotechnology. Applications of biotechnology including producing a variety of proteins from bacteria and fungi, such as when we produce vaccines or enzymes in a microbe. That is genetic engineering. I'm trying to think if the new vaccine of COVID-19 was made in microorganisms, and I don't know. I knew the no vaccine, at least Pfizer and Moderna, is using messenger RNA, but I don't know how they made the vaccine, so can't tell you. Gene therapy is another application of biotechnology. Gene therapy is when a baby is born and he has a defective gene, and then they put in the correct gene, or they add the correct gene which complements the mutated gene, and then the patient has gene therapy which corrects their phenotype. It corrects the patient's some signs and symptoms. The third application of biotechnology is when we genetically modify bacteria to produce something that we want, such as we've added, I just lost my screen there for a moment. We've added something to bacteria to then coat plants to protect the plant from insects or from freezing. And you don't have to add it to the bacteria. You could genetically modify the plant directly, the plant cell itself, and like put in a natural insecticide that the plant will make, and then help protect the crop or the plant from insects. Any question about any of that? All right. Microbes are also important in human welfare. In microbial ecology, that is where bacteria are doing something in the ecosystem, such as recycling carbon, nitrogen, nutrients, sulfur, and phosphorus. And they recycle these elements or nutrients so that they may be used by plants and animals. So microbial ecology is where these elements and nutrients move around in the ecosystem so that they may become available for other organisms, such as plants or animals. We've talked about the nitrogen cycle, which is a cycle of microbial ecology where nitrogen moves around in the ecosystem because of the microbes. And this is an example of a microbial-mediated biochemical cycle that help support life on Earth. Microbial ecology, as well as the microbial-mediated biochemical cycles, were first studied by Winogradsky and Bajering, and they were the first to show the recycling of nutrients by bacteria in the ecosystem. We'll talk a little bit about Winogradsky later. He was different from all other microbiologists who they studied pure cultures, meaning one species of microorganism, to try and understand it. Winogradsky was studying many different microbes all together, and that is why he was able to see the recycling of nutrients in the ecosystem, because like when we're talking about the recycling of nitrogen on the planet Earth, one microbe will move it from one position, and then another microbe will move it from that position to another position. And so to study the biochemical cycles on Earth, you actually had to study many microbes, and that is exactly what Winogradsky did. Any question about any of that? All right, no questions. Let's move on. Look at another example of how microbes help human welfare. Microbes are used in bioremediation. Bioremediation means literally life and to remedy. And this is where we use living organisms to deal with an environmental problem. For example, these individuals are spraying microbes on an oil spill to get the microbes, especially the bacteria, to digest the pollutants. And we've already talked about how microbes digest our sewage. Well, when we have pollutants like an oil spill, it is the microorganisms that digest the oil, converting the oil into CO2 and water. We can also use bacteria to bioremedy other pollutants like mercury. We can convert mercury to a less toxic form by using bacteria. Any question about any of that? So the point is, microorganisms can degrade or detoxify pollutants, breaking them down into less harmful substances. Microbes can also be used as an alternative to an insecticide. We call these biological insecticides. There are certain microbes that are pathogenic to insects, and they would be an alternative to a chemical pesticide. A microbe that is pathogenic to insects. That would prevent insect damage to crops, as well as prevent disease transmission. An example is Bacillus thuringiensis. An infection by Bacillus thuringiensis is fatal to many insects, and yet Bacillus thuringiensis is harmless to humans and other animals and plants. My father actually used Bacillus thuringiensis way back in the 1960s. He coated his apples with this bacteria to prevent the apples from getting worms. Okay? The trouble is, he was in Western Oregon or Western Washington, and it rains a lot. And he didn't know after it rains, you need to treat the apples again. And also you need to apply it about every 2 weeks, which I think was on the package. But he was a bit lazy, and he did it once or twice. And it worked for a while, but you need to repeat the treatment about every 2 weeks, or more often if it rains, as the rain washes the Bacillus thuringiensis off the apples. All right, any question about that? If there's no questions, we're going to move on to another topic, the naming and classing of microorganisms. And the scientific names were started by this guy here. Does anybody know who this is? Actually, we talked about him yesterday. Okay, the scientist who first started naming as well as classifying all organisms, especially microorganisms, was Carolus Linnaeus, and he liked Latin so much he Latinized his name. I think if you go and look at it, he's Scandinavian. I don't remember which country, maybe Sweden. It was Carl Linnaeus, and usually it's pronounced Carl Linnaeus, but he even Latinized his first name, Carolus Linnaeus. And he established the system for scientific nomenclature, how we name things. It's also called the Binomial Nomenclature because all species names have 2 names to them. Each organism has a genus name and a specific epithet, which is roughly the species name. The species name is actually the 2 names, Homo for humans, which is the genus, and then sapiens, which means wise man, which is the specific epithet. And then the species is the 2 names, Homo sapiens. So that's why it's called the binomial nomenclature, because it has 2 names, but it's also called the scientific nomenclature. And it's used worldwide, so you could go to any country in the world and say Homo sapien, and somebody who knows science would know what that was. Any question about that? Anyways, it was such a good system, we're still using it today. The names are either italicized, like if you print it on a computer, or if you're like writing it on the board, you underline it. So italicized or underlined. All scientific names are italicized or underlined. The genus name is always capitalized, and the specific epithet is always in lowercase. So if we go backwards, Homo is capitalized, and then sapiens is always lowercase. The scientific names are all Latinized, and they're used worldwide. The name may be descriptive, like Homo sapien means man who's wise, or wise man. Okay? But the name can also honor a scientist, like Escherichia coli. Escherichia is honoring a early microbiologist who I think was Escher. And then the coli part is descriptive. You can find E. coli, Escherichia coli, in the colon, and so that's what coli stands for. So Homo sapien is descriptive, And Staphylococcus aureus is also descriptive. It literally means a round cell, cocci, that grows as a grape cluster, staphylococci, and aureus means it's golden, so it means golden grape cluster cocci, or round cell. Okay, so totally descriptive. Any question about any of that? After first use, scientific names may be abbreviated with the first letter of the genus and a period, and then the specific epithet. Like you say Staphylococcus aureus the first time, and then all other times you're using it, you say S. aureus. And Staphylococcus aureus is found on the skin of people, as well as the nasal and throat passages. And you say Escherichia coli, like if you're writing it. And then all other times you can say E. coli, which is found in the large intestine. Now, E. coli is so common that even scientists generally call it E. coli, meaning the first time they don't say Escherichia coli. And that's because it is so well known. Generally, most bacteria are not so well known, but E. coli is. So if you say E. coli, it won't correct you. In a scientific paper, they do tend to write Escherichia coli the first time, and then E. coli all other times. But that's about the only time even scientists use Escherichia. All right, so when we're classifying microorganisms, before we address this topic, we first need to take a look at the classification of all living organisms on the planet Earth. So when we're classifying microorganisms, or actually any organism, we divide all organisms into the first division, which is the three-domain system. So we have all life on the planet Earth, and by the way, this is from Chapter 10. And then we divide all living things on the planet Earth into the three-domain system. The three-domain system, if you were to graph it, kind of branches, sort of like a tree, so they call it the tree of life. The three-domain system has three domains: domain bacteria, domain Archaea, and domain Eukarya. The domains are classified based upon their genetic data. What we do is we sequence the DNA in an organism and then look to see where that organism falls in the domains. So all bacteria have a sequence of DNA which is similar to each other and more similar to any organism in another domain. So all of the bacteria have a genetic sequence similar to each other and more similar to any other organism in another domain. And that is how we classify the domains. It's based on genetic data. This came as a surprise to, well, actually all biologists, and that is when they sequenced living organism that we had three domains, because biologists thought that the bacteria and the archaea were similar to each other. And they thought that the bacteria and the archaea would have been in one group, which they called prokaryotes, and the other group they called eukaryotes. They knew the eukaryotes were different from the prokaryotes. They did not know the bacteria were different from the archaea, or I should say, we did not know that they were very different from each other. And in fact, the bacteria are actually a little more different from archaea than the archaea are from eukarya. And this is a very slight difference, but the archaea are a little bit closer to the eukarya than they are to the bacteria. And we're talking about a slight difference here, meaning generally speaking, all three of them are their separate domains. The point is that each domain has a different sequence of DNA. When we're talking about the domains, the domain Eukarya, we will break into different kingdoms. The domain Bacteria does have a kingdom, but it's only one kingdom. So the domain Bacteria is also the kingdom Bacteria. And we're not going to talk about that. And the same with Archaea. It has a kingdom, and it's simply the Archaea kingdom. The domain Eukarya, though, has different kingdoms. And we're going to use the six-kingdom approach, meaning kingdom Bacteria, kingdom Archaea, kingdom Plantae, kingdom Animalia, kingdom Fungi, and kingdom Protista. Okay? The kingdoms, do I have that on this slide or the next slide? I hope it's this slide. The kingdoms do not have a defining characteristic, meaning we have no definition for what a kingdom is. So there's an argument about which organism falls into which kingdom, as well as how many kingdoms there are. We're going to use the six-kingdom approach, but there are other kingdom approaches. There are 5-kingdom approaches, 6-kingdom approaches, 9-kingdom approaches, 20-kingdom approaches, and then higher number kingdom approaches. And like I said, we're going to use 6. I think everyone's familiar with plants, animals, fungi. You may not be familiar with Protista, and hopefully you will become familiar with Archaea and bacteria. So we'll use the 6-kingdom approach, which I think is the approach your textbook author is using. We will hardly talk about kingdoms at all, but we will briefly talk about plants and animals. Any question about the 3 domains of life or the kingdoms? Remember, the domains of life are defined. The kingdoms are not. And I may not have said it directly, but scientists argue among themselves what is a kingdom, which organism belongs in which kingdom, and how many kingdoms there are. So there is no agreement among scientists among any of that. And the reason is because there is no definition for what a kingdom is. And we won't look into that argument. All right, let's talk a little bit more about the three-domain system. We have different cell types within the three domains. And a cell type is something your author is calling it. You could call this a super kingdom, meaning bacteria have the cell type prokaryotic cells. And that is a cell type. It is also a super kingdom, but your textbook author doesn't use the term super kingdom. Archaea also has the cell type prokaryotes, meaning all the cells of the Archaea are prokaryotic cells. And we'll talk a little bit more about prokaryotes and eukaryotes later in the term. The domain Eukarya have eukaryotic cells, so their cell type is eukaryotic. Any question about that? All right, no questions, let's move on. And we're going to talk a little bit about the characteristics of the different domains, starting with the domain bacteria. The bacteria have their cell types prokaryotes. The bacteria have peptidoglycan in their cell walls. Peptidoglycan is a specific molecule, and we'll talk a little bit more about that later. But we can always find peptidoglycan in the cell walls of the bacteria. The bacteria mostly multiply by binary fission. Binary fission is the cell simply splits into two. The bacteria multiply by binary fission. For energy use, the bacteria can get their energy needs from all of the possible sources. They can get their energy needs from an organic chemical. And that's where we get our energy needs from. From the glucose we eat, as well as the other food molecules that we eat. And for us couch potatoes, we usually break the food down and then break it down into glucose and use it that way. And we'll talk a little bit about that. Anyways, bacteria can get their energy needs from organic chemicals, but some bacteria can get their energy needs from an inorganic chemical. And that would be something like hydrogen sulfide, or ammonia, or sulfate. And then some of the bacteria can get their energy need from light. And these are the photosynthetic bacteria. So the bacteria can get their energy needs from organic chemical, an inorganic chemical, and from photosynthesis. Most bacteria are free-living decomposers or primary producers. A decomposer is something that decomposes things. So it causes the tree to rot, for example, and decompose. A primary producer is something that makes food in the ecosystem. So most bacteria are free-living decomposers or primary producers. Some bacteria are parasitic. A parasite is something that lives off of something else. So some bacteria live off of something else. And some bacteria are pathogenic, meaning that some bacteria can cause disease in humans. Pathogenic, cause disease in humans. Any question about any of that? All right, let's talk a little bit about the characteristics of the Archaea. The Archaea all have prokaryotic cells, so their cell type is prokaryote. Does anybody know, do we find peptidoglycan in the cell wall of Archaea?
No.
Yes, you're correct. Peptidoglycan is only found in the cell walls of bacteria. Some of the Archaea are found in extreme environments. So some are in extreme environments. These include the methanogens. Some organisms that produce methane gas are Archaea. Some of the Archaea are found in extreme salt conditions, like the Dead Sea between Jordan and Palestine is 30% sodium chloride, and only extreme halophiles can live in that. A halophile is something that loves salt. Halo, salt, phile, lover. There are some Archaea that are extreme thermophiles, meaning they live at elevated temperatures. And some of them can live at higher temperatures than boiling water. Most extreme thermophiles live around 95 degrees Celsius. 100 degrees Celsius is when water boils. But if we look at all of the Earth's biomes, we will find Archaea everywhere on the planet. So the Archaea are found more than just in extreme environments. They're found everywhere. Does anybody know, are the Archaea pathogenic to man? Does the Archaea cause disease in humans?
No.
Correct. The Archaea do not cause disease in humans. And for this reason, we were not going to study the Archaea much after this lesson. Meaning in lesson 1 we'll talk about the Archaea, but after lesson 1 we really won't study the Archaea because they do not cause disease in humans. And the focus of this class is to talk about disease-causing organisms. And guess what? All of the disease-causing bacteria are in the domain bacteria. And I didn't say that correctly. All of the disease-causing prokaryotic cells are in the domain bacteria. Meaning I use bacteria to say prokaryotic cell the first time, and then I use bacteria to mean the domain bacteria. And I'll try not to do that. When I use the word bacteria, I will mean the domain bacteria. Any question about any of that?
You said that all the disease-causing bacteria are in the domain bacteria?
Yes, all of the disease-causing prokaryotic cells are in the domain bacteria. Okay? There are some disease-causing eukaryotes, and we will talk about them later in the term. like an amoeba that causes amoebiasis, like histolytica into amoeba, causing diarrhea. That is not a bacteria. That is a eukaryotic cell. So there are some eukaryotes which cause disease in humans. Okay, but of the prokaryotic cell, all of the prokaryotic cells that cause disease in human are in the domain bacteria. None are in the domain Archaea. All right, let's briefly talk about some of the characteristics of the Eukarya. We divide the Eukarya into 4 different kingdoms: Plantae, Animaliae, Fungi, and Protista. So the plants, the animals, the fungus, and the Protists. When we're talking about the Protists, you should realize that the kingdom Protista is a collection of all the eukaryotes that are not plants, animals, or fungus. So this kingdom is a little different in that it is not monophyletic. It is a collection of all the eukaryotes which are not a plant, an animal, or fungi, meaning the Protista are not similar to each other. And many people, if they have more than 6 kingdoms, they will divide the Protista into many different kingdoms. Okay? For the eukaryotes, all of them lack peptidoglycan in their cell wall. And of course, animals don't have a cell wall, but all eukaryotes lack peptidoglycan. So this molecule is only found in the domain bacteria. And it's found in the cell wall of bacteria. Let's talk a little bit about the kingdom fungi. Fungi is plural. If you're talking about a singular fungus, you use the term fungus. They have chitin in their cell walls. You don't really need to know that. But they use organic chemicals for energy. So they are like us in that the fungi can only use organic chemicals for the deriving their energy needs. There are multicellular fungi, like the molds and the mushrooms, and they consist of masses of mycelia, which make up the mushroom or the mold. And these mycelia are composed of filaments called hyphae, and we're looking at that here. But most of the fungi are not multicellular. They are unicellular. All of the yeasts are unicellular. The fungi are all decomposers or pathogens. A decomposer decomposes something that died. A pathogen causes disease. All right, any question about the fungi? If not, let's move on to the kingdom Protista, the Protists. The Protists have 3 different categories. The algae, which are plant-like protists, the protozoa, which are animal-like protists, and then the third group, the fungus-like protists. The algae engage in photosynthesis, and that's why we call them plant-like. So they make their own food. They also have cellulose in their cell walls, so that's similar to plants. Like I said, they engage in photosynthesis. They produce oxygen from photosynthesis, and they also produce glucose and other organic compounds. So the algae are primary producers on the planet, and they engage in photosynthesis. We're looking at an algae here. It's a colonial algae made up of single cells, and you see each dot here? That is a cell, and this is a colony, and this parent colony has made 6 baby colonies, and this will open up and release the baby colonies to go elsewhere to form their own colonies. The protozoa are the animal-like protists, and they are called animal-like because of their needing to eat an organic chemical like animals do, as well as many of them move, and animals move. And some examples of the protozoa include amoeba, paramecium, and euglena. They may be motile, meaning they move, with pseudopods, with cilia, or with flagella. This is the pseudopod, and that looks like a white, no, that's an amoeba, sorry. Some of the protozoa are parasites and cause disease in people. I mean, they can cause disease in other organisms too, but we're talking about people. And remember, the third group are fungus-like protists. They decompose things, decompose dead things. All right, any question about the protists? Remember, they are not monophyletic. If you were to sequence the algae and sequence the amoeba, their DNA would be very different. And the amoeba would be closer to animals than they would be to the algae. And the algae would be closer in their sequence to the plants than they are to the amoebas. So the protists are a collection of all the eukaryotes, which are not plants, animals, or fungi. All right, if there's no question, let's move to the last 2 kingdoms, the kingdom plants. We can divide the kingdom plants into mosses, ferns, conifers, plants that produce cones and produce seeds in the cones, and then the flowering plants, the plants that produce flowers to get their seeds. All of the plants are multicellular. We're not going to be studying plants in this class because no plant causes disease in humans. Any question about that? No plant causes disease in humans, and no plant causes disease in animals. Any question about plants?
Isn't there like certain like flowers that could be deadly to humans?
Yes, that would be a toxin, but that doesn't cause disease in the human. I'm not saying that plants aren't poisonous, okay? And their poisons can kill humans. I'm just saying the plant won't grow in a human and then cause disease. And you could argue that that's a fine distinction. I'm not going to go there, okay? Yes, plants can be poisonous, and if you eat the plant and it's poisonous, it can kill you. But, I don't know. That's not really a disease. That's from consuming a toxin. Okay? All right, let's move on to kingdom animal. You can break the animals into sponges. The worms, which the scientific name is a helminth, the insects, and the vertebrates. All of the animals are multicellular. So none of the plants and none of the animals are single cellular. Any question about that? The animals, we will be studying some of them later. Some of the worms cause human disease. An example would be a tapeworm. And these worms are parasites. And so worms or helminths can cause disease in humans. Any question about any of that? All right. If there's no questions, it's now after 2 o'clock. This would be a good time to stop, and let's come back at 2:30. Okay? All right, any questions? So we're going to take a break, go grab something to eat or something to drink, and then come back at 2:30. If you want to know why I want a long break, it's because I'm not sure how Panapto will handle a short break, and I want it to be long enough so that it'll end, and then I can restart it.
Okay, thank you.
All right, I'll see you at 2:30.
Bye-bye.
Thank you.
All right.